In the fertile lands of a small farming community, nestled between rolling hills and picturesque landscapes, the dedicated farmers toiled day and night, cultivating crops that would nourish their families and sustain their livelihoods. Among the many crops they grew, Brussels sprouts stood out for their nutritional value and unique taste, but their cultivation was not without its challenges. Year after year, a pest called the cabbage moth wreaked havoc on the Brussels sprout fields. The moth laid its eggs on the tender leaves, and when the caterpillars emerged, they voraciously feasted on the precious foliage, leaving behind devastation in their wake. The farmers tried their best to protect their crops, resorting to chemical pesticides, but the long-term consequences on the environment weighed heavy on their hearts. As news spread about the detrimental effects of pesticides and the growing demand for safer and more sustainable food options, a team of passionate scientists and researchers took it upon themselves to find a solution to the cabbage moth problem. They sought an eco-friendly approach that would safeguard the crops while preserving the delicate balance of nature. Their quest led them to the fascinating world of genetic modification. The researchers delved into the genetic makeup of various plant species related to Brussels sprouts, searching for natural resistance to cabbage moths. After rigorous study, they unearthed the key, specific genes that produced insecticidal proteins capable of warding off the dreaded caterpillars. With newfound hope, the scientists embarked on a journey of genetic engineering. In state-of-the-art laboratories, they carefully isolated and amplified the resistant genes, transforming them into a form that could be integrated into the Brussels sprout genome. They called it the Resistagreen project, a symbol of their determination to create a pest-resistant variety. Through a meticulous process of gene insertion, the researchers successfully incorporated the insecticidal genes into the Brussels sprout plants. But they knew they couldn't stop there. The team understood the importance of controlling the expression of these genes, allowing the insecticidal proteins to be produced only when needed, when the cabbage moth larva attempted to devour the leaves. It was a delicate dance of genetic engineering and regulatory elements, a symphony of science that would harmonize resistance and ecological balance. After years of hard work, dedication, and numerous trials, the first generation of Resistagreen Brussels sprouts emerged from the laboratories and found their way into the test fields. Farmers eagerly welcomed these resilient plants into their farms, hoping for a natural defense against the cabbage moth invaders. Under the watchful eyes of both scientists and farmers, the GM Brussels sprouts grew tall and strong, their leaves showcasing newfound resistance. The results were astounding. The Brussels sprouts stood tall, undeterred by the cabbage moth caterpillars that had once threatened their existence. The insecticidal proteins kept the pests at bay, preserving the integrity of the crops and ensuring a bountiful harvest. With this triumph, the Resistagreen Brussels sprouts became a beacon of hope for sustainable agriculture. Farmers reduced their reliance on harmful pesticides, embracing a more eco-friendly way of farming. The market embraced the GM Brussels sprouts, offering consumers a healthier option free from the worries of pesticide residues. The success of the Resistagreen project inspired more research and innovation in the world of genetic modification. The farmers' community and consumers alike looked forward to a future where resilient crops and a greener planet coexisted in harmony. And so, in the fields of that small farming community, a story of determination, ingenuity, and a shared vision unfolded. The tale of cabbage moth-resistant Brussels sprouts proved that science, when wielded responsibly and with care for nature, could yield solutions that nourished both body and soul.